Get ready to explore a fascinating topic in genetics. In this video, we'll delve into the intriguing world of contiguous gene deletion syndromes. You'll discover how these syndromes result from deletions of large segments of DNA containing multiple genes, leading to complex and varied symptoms. We'll dive into specific examples, such as WAGR syndrome, Giedion syndrome, 1p36 deletion syndrome, Azoospermia, and Miller-Dieker lissencephaly syndrome, and explore how these conditions can impact an individual's life. So sit back, relax, and prepare to embark on an exciting journey into the world of contiguous gene deletion syndromes. So, first is WAGR syndrome. Frequency of WAGR syndrome is 1 out of 10,000. Genetics of WAGR syndrome is that it is contiguous gene deletion syndrome, and it is del 11, p13, which means deletion on chromosome 11 at small arm p at band 13. Moving to features of WAGR syndrome, and that are Wilms, or embryonal renal tumor. Wilms tumor, also called Wilms tumor or nephroblastoma, it is a type of childhood cancer that starts in the kidneys. Aniridia. Aniridia is an eye disorder characterized by a complete or partial absence of the colored part of the eye, which is the iris. These iris abnormalities may cause the pupils to be abnormal or misshapen. Aniridia can cause reduction in the sharpness of vision and increased sensitivity to light. Genitourinary abnormalities, including gonadal tumors, and retardation of growth and development. Moving into further details, to discuss the basic etiology of WAGR syndrome. This syndrome is caused by loss of gene paired box 6. Paired box 6 protein is involved in the development of a specialized group of brain cells that process smell. Loss of Wilms tumor suppressor 1. Wilms tumor suppressor 1 is a transcription factor that plays a role in genitourinary organ development. And loss of brain-derived neurotropin factor. Moving to management of WAGR syndrome. So, Wilms tumor gene 1 DNA probe can be used for antenatal diagnosis. So, this is chromosome 11, showing region deleted in WAGR syndrome, which are brain-derived neurotropin factor, paired box 6, and Wilms tumor suppressor 11. You can memorize this syndrome by linking all point. Let's link them. W for Wilms tumor, A for aniridia, G for genitourinary abnormalities, and R for retardation of growth and development, hence called as WAGR. WAGR syndrome is loss of gene paired box 6 that confers aniridia. Loss of Wilms tumor suppressor 1 that causes Wilms tumor. Loss of brain-derived neurotropin factor relates to retardation. Moving to next contiguous gene deletion syndromes, and that's Giedion syndrome. Frequency of Giedion syndrome is rare. Genetics of Giedion syndrome is that it is contiguous gene syndrome. It is del 8 q24.11 q24.13, which means deletion on chromosome 8 at long arm q between band 24.11 and 24.13. It is autosomal dominant inheritance. Features of Giedion syndrome are large, laterally protruding ears, elongated upper lip, bulbous nose, sparse scalp hair, short fingers and toes, winged scapulae. The scapula, or shoulder blade, is the bone that connects the clavicle to the humerus. The scapula forms the posterior of the shoulder girdle. It is a sturdy, flat, triangular bone. The scapula provides attachment to several groups of muscles. Multiple exostoses, which are tumorous bony overgrowths, redundant skin, and intellectual handicap. Moving into further details, let's discuss the etiology of Giedion syndrome. Let's understand the normal pathway first. protein exostosin 1 normally forms a complex with exostosin 2 that catalyze polymerization of heparin sulfate. The complex is an essential factor in a signal transduction cascade that regulates chondrocyte differentiation, ossification, and apoptosis. Langerhans-Giedion syndrome is caused due to mutations of exostosin 1. You can memorize Langerhans-Giedion syndrome as exostoses, which is tumorous bony overgrowths in Langerhans-Giedion syndrome, and exostosin 1 mutation in Langerhans-Giedion syndrome. Moving to next, contiguous-gene deletion syndromes. And that's 1p36 deletion syndrome. Frequency of 1p36 deletion syndrome is 1 out of 5,000 live births. It is the most common terminal deletion syndrome. Genetics of 1p36 deletion syndrome is that it is contiguous gene syndrome. It is del 1p36, which means deletion on chromosome 1 at small arm p at band 36. Etiology of 1p36 deletion syndrome is defects due to haploinsufficiency of several genes near the 1p terminus. Let's discuss features of 1p36 deletion syndrome. Small head, unusually short and wide, straight eyebrows, sunken eyes, midface hypoplasia with flat nasal bridge, asymmetric ears, pointed chin, hypotonia. Hypotonia is a term that describes decreased muscle tone. Typically, muscles have a very small amount of contraction that gives them a springy feel even when relaxed. Developmental delay, severe learning difficulties, epilepsy, patent ductus arteriosus, and other cardiomyopathy. Hearing impairment, hypermetropia. Longsightedness, also known as hypermetropia or hyperopia, is a common vision disorder. It results from the overall power of the eye not being strong enough or the length of the eye being too short. Moving to next, contiguous gene deletion syndromes. And that's azoospermia. Frequency of azoospermia is. This deletion may be responsible for 10 to 20% of cases of idiopathic means occurring without apparent extrinsic cause, azoospermia, and severe oligospermia, which means complete lack or severe deficiency of sperm. Oligospermia means that you do have a measurable amount of sperm in your semen, but the numbers are lower than the typical numbers. If you have azoospermia, it means there are no sperm seen in your semen. Genetics of azoospermia is that it is microdeletion, del y 11.2, which means deletion on chromosome Y at band 11.2. Feature of azoospermia is male infertility. Moving into further details of etiology of azoospermia. Y chromosome has P arm and Q arm. There is SRY region on P arm, which stands for sex-determining region Y gene. It is found on the Y chromosome. In azoospermia, deletions cause loss of genes within the azoospermic factor region, including 3 subregions, which are azoospermia factor region A, B, and C. These subregions are responsible for different phases of spermatogenesis. You can memorize important points about azoospermia by memorizing that azoospermia is the medical term used when there are no sperm in the ejaculate. It can be obstructive, where there is a blockage preventing sperm from entering the ejaculate, or it can be non-obstructive, when it is due to decreased sperm production by the testis. Sperms are produced by male, and Y chromosome is responsible for maleness. So, there must be defect in Y chromosome in azoospermia. Moving to next, contiguous gene deletion syndromes. And that's Miller-Dieker lissencephaly syndrome. Frequency of Miller-Dieker lissencephaly syndrome is rare. Genetics of Miller-Dieker lissencephaly syndrome is that Miller-Dieker lissencephaly syndrome is Autosomal recessive inheritance. Autosomal recessive inheritance is a way a genetic trait or condition can be passed down from parent to child. A genetic condition can occur when the child inherits one copy of a mutated gene from each parent. The parents of a child with an autosomal recessive condition usually do not have the condition. It is del 17, p13.3. Which means deletion on chromosome 17 at small on p at band 13.3. So etiology of Miller-Dieker lissencephaly syndrome is haploinsufficiency of one or more genes at 17p13.3. So, it is visible or submicroscopic contiguous gene deletion. Del 17, p13.3. Moving towards features of Miller-Dieker lissencephaly syndrome. Classic lissencephaly smooth brain. Subtle brain defects include microcephaly, prominent occiput, narrow forehead, upslanting palpebral fissures, small nose and chin, cardiac malformations, hypoplastic male genitalia, macrosomia, mild developmental delay, severe intellectual handicap, seizures, attention deficit hyperactivity disorder, psychomotor retardation, and death usually in early childhood. Management of Miller-Dieker lissencephaly syndrome is sometimes correction of omphalocele or umbilical hernia. Attention to feeding problems. So, this was all. In our next video, we will talk about the single gene syndromes.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.